Welcome and thank you so much for joining us. My name is Caitlin O'Brien and I'll be moderating with my co-host Lindsay Feistumo. Today we're going to be meeting our fisheries crew from the Patrick Center for Environmental Research. We're going to be able to see them in the field, and then we're going to turn to all of you to discuss their field work and learn a little bit more about what they do. Before we get started, we do have to go through some housekeeping just to make sure you, our members, know how to ask questions and participate with us. So if you've never joined us before, and if you don't have video or microphone capabilities, you'll want to type your questions into the chat window. You'll see the icon to open the chat at the bottom of your screen. Also at the bottom of your screen is the participants panel. If you would like to ask a question directly and have audio and video functions, please open the participants panel and click the raise hand button. That will let us know you'd like to ask a question, and we'll be able to unmute your microphone for you. Now, let's get started. While you'll have the opportunity to see many of our fisheries crew field in action in the field, fisheries biologists Dave Keller and Allison Stoloska will be joining us today for a discussion after the field work video, which is very exciting. Dave and Allison, we cannot wait to chat with the both of you. Dave and Allison coordinate fish data collection, analysis, and interpretation. Their work takes them into streams and rivers all over the country, where they catch, identify, and release freshwater fish to study population dynamics and water quality issues. Let's tune in to see them in action.
My name's Dave Keller, and I am a fisheries biologist at the Academy of Natural Sciences. In the fisheries department, we survey fishes to assess their distribution and abundance.
Cool. Don't know why that stopped, sorry.
And to assess the impacts of human beings on fish. Today we are at Kirkwood Preserve on Crum Creek. We're near the headwaters of Crum Creek, which is a tributary to the Delaware River. We're about 30-40 minutes from Center City, Philadelphia. So today we had a large crew, the fisheries department, Dr. Horowitz, Paul Overbeck, Allison Stocklosa, fisheries biologist from the Academy. We also had folks from the Riverkeeper and Willistown Trust, all here to survey the stream today to assess the fish community and the American eel that lives in the stream. So we're using a backpack electrofishing rig. This is a backpack that has a small power source on the back of it that we then use to put some electricity into the stream. When a little bit of electricity goes into the water, this temporarily immobilizes the fish. They typically roll over and float to the surface, or are carried by the current downstream. And it's at that time that we net them and we'll place them into a bucket. And normally they revive within seconds.
See anything?
Not yet. Oh, something straight ahead, like 5 feet ahead of you. Floating on the surface.
They're all hiding under the stump.
Yep, we're getting a lot of fish right here, like Allison just said, under the stump. This is good fish habitat, providing some cover. One thing commonly that we deal with at this time of year is little willow leaves that drop themselves in the water, and they look just like a minnow. And we commonly net them, and
the acorns today are looking like crayfish. I see something happen. Did you see it come out?
Yeah, I think it went up this way. Picking up a fair number of, yeah, like kind of your lentic species, your pond species.
I feel like that's how it was at the other sites.
We're not able to get every fish, but we're getting a sample. So it's representative of what is here. Whoa, 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 whoa. Whoa. We got this guy. I think we got this last year. Look at this.
Huge snapping turtle.
Big snapping turtle. I'll tell you what, I don't want it to tear up my net, so let's see if we can get them into the shallows. Does anyone feel like picking them up? Or no?
So he came out of the root wad.
I'm going to let him go. If anyone else wants to. All right.
Yeah, it smell like snapping turtle for a while.
Once we've gone through the stream and collected the fish, we'll then come to a location where we'll identify the fish and measure the fish, recording information on their length, noting any abnormalities or lesions or evidence of any disease. X max 3.7. No ins 3.8. No, that was a no ins at 3.8. You want them for the small board, right? Oh, okay, got you.
Shake it down.
Sometimes they'll cooperate. Oh, there you go, there you go. 27.1.
Went right through that hole. Quiet down.
72.
It looks like
72.4.
okay. That's supposed.
Wow. That's bigger than
72.4 was what we got.
and you got a 60 something last
We got
year.
69 last year, so
Might be.
lime.
More?
After we identify and measure the fish, we then return them to the stream. Today we found many fish, different species of catfish, American eel, different minnow species, different sunfish species, largemouth bass, and sunfish. This work is important to assess the impacts of American eel on aquatic communities. We're hypothesizing that American eel presence has an impact or an influence on crayfish communities by preventing or serving as a type of biotic resistance against invasive crayfish species. Fish are important constituents of streams because they integrate the activities of lower level organisms that are in the stream. Algae, macroinvertebrates, and fish are generally at the higher ends of the food chain. So surveying fishes can tell us a lot about what is going on in the ecosystem.
Fantastic. Thank you so much, Dave and Allison. I'm going to go ahead and stop sharing my screen so we can go ahead and turn it over to hear from them. We'll keep an eye out for raised hands and questions in the chat. But first, is there anything you wanted to add, Dave and Allison, about what we just saw in the video?
No, I'm curious to hear what questions from the audience. The video was a little choppy on my end. I may have had a bad connection, but curious to hear what some folks say. I see that someone has asked, does electrofishing stun reptiles too? And the answer to that is yes. We do encounter snakes and turtles and frogs and salamanders, and they do also get stunned by the electrofishing equipment.
Great. We have another question also a little bit earlier in the chat. Cindy lives in the Susquehanna watershed and we need fish lifts to help fish over the dam. Can eels use the fish lifts to get over the dam?
Yeah, absolutely. The lifts are there to help a lot of migratory fish, not just eels, but also American shad and other fish that are trying to move upriver but are stopped by the dam. So.
Excellent. So we're not seeing questions anymore right now at the moment. Allison and Dave, do you want me to show some of the other fish photos that we have about going out in the field, and we can maybe talk through those?
Sure.
Okay, great. Give me just a second and then while I get this set up. But we're really grateful to all of our members that were able to join us today. It is a beautiful fall afternoon. So thank you so much for joining us. So can everybody see the backpack electrofishing photos? Excellent. Allison, do you want to sort of take us through sort of what we saw in the video and what we're seeing in these photos?
Sure. So this is kind of just an example of some of our backpack electrofishing, which you saw us do in the video. But you can kind of see the gear setup. Um, you can see how we kind of move through the streams almost in a line. You know, for a lot of our surveys, we're really trying to assess the population. So we want to try to get all of the fish that are in the reach of the stream that we're sampling. So we kind of move in this line-like fashion that you can see there. And down in the bottom corner is another technique that we use with the backpacks where we're sampling a known area. So that's a 5 meter by 5 meter square. And the fish are going with the current into the same block net that we're holding down there. So just a couple of different techniques based on what our questions that we're trying to answer are. And all of this work that we're doing, you know, for the most part, we're trying to do it in a quantitative way so that we can really make some statements about the fish and what's going on in our streams. So that's another reason why we have, you know, these known areas or this known section of stream that we're sampling, so we can really tell how many fish per meter or how many fish per that section of stream that we're sampling.
Nice. And then could you talk a little bit about what you're doing if you're on a boat and electrofishing?
Sure.
Or Dave, either one of you.
Allison.
So these are just some other examples of our other techniques that we do. You know, a lot of our work revolves around electrofishing. So we have a couple different boats that we use, and it's the same idea. Some currents put into the water. The fish flip over and float up, and we catch them, and you can kind of see in that top one that we put them in recovery bins. It's almost like tasing the fish is what it's the most similar to. So as soon as the electricity stops being applied to the fish, they revive in those tins, which we're able to then measure, identify them, and put them back where we found them at the end of the day. And we also do seining, which is just those big nets, and you pretty much just pull them through different areas of the water, and you catch all the fish that way too. So these are just a couple examples of some of the other things we do. Dave, do you have anything to add about our techniques or?
No, but I just saw two good questions. One from Dan, and then another question that kind of was building on the Susquehanna conversation. And I real quickly to respond to Dan's question about have we noticed any changes on Crum Creek? I'd say, you know, we haven't in my time. I see another question here. Well, see that. So it's interesting. Cindy's asking, asking a question about how often we visit a site, and so that kind of plays into my answer to Dan about Crum Creek. You know, we don't always visit Crum Creek every year, and it depends on the questions that that we're trying to answer. But I can say over my time at the Academy, roughly 15, 16 years being here at the Academy, that you know, we haven't noticed any major changes on Crum Creek, but I think that as you have blockages that come out on Crum Creek, so you have small dams that are removed, that increases connectivity, it increases passage. As you get to the upper portions of Crum Creek, there's groups like the Willistown Trust that we've been talking to and other groups are working with, and there's other groups outside of Willistown Trust that are also working in that area that are working to improve and build on the positive things that are there in Crum Creek. So some nice cold, there's some evidence or some pockets of cold groundwater, spring-fed areas that support salmonids, support trout. And so there's some interest in building on that and restoring, conserving what's there and restoring trout populations to some of the upper headwater portions of Crum Creek. So those are a couple comments about Crum Creek. And then the other thing I'd like to come back to on the Susquehanna, someone mentioned snakeheads being a component of the decision making in terms of whether to operate that lift on the Susquehanna. And that's something that we encounter, and Allison and I and others in the fisheries section, we commonly encounter these kinds of competing interests. So when you talk about fish and fish management, you often can't just limit it to one specific species. What's best, let's say, for the Susquehanna to restore American eel, American shad, and other migratory fishes would be to remove the dam or increase passage. But at the same time, we have an invasive species, the northern snakehead, that can cause a lot of problems for the ecosystem upstream. And so managers have to balance the two, come to and make a decision. And that's where our work comes in. That's what Allison and I and others in the fisheries department really work on, trying to answer some of these questions so that we can better manage the resource that we have.
Excellent.
And I'm just looking through the chat here. I see. Yeah.
We have a question. Oh, I was just going to say we have a question. Do you check pollution levels in the sites?
The short answer is yes. I didn't know if Allison, if you wanted, did you have any thoughts on that? If not, I can answer that question.
Yeah, I mean, I think it depends on exactly the project that we're working on, what our goals of that project are. So certain things about fish assemblages can kind of tell us the water quality of the sites. We can tell based on how many fish and what species are there, kind of the health of the system. And we also do have projects where we're looking at specific contaminants. So in those projects, we would bring back some fish and analyze the tissue for things like PCBs, mercury, whatever toxins that we're really concerned with at that time. So it's all kind of based on the questions that we're trying to answer and the systems that we're working in.
Yeah, and I'll add to that too that Allison mentioned it. She mentioned assessing the fish assemblage. So do we check for pollution? What lives in the stream can tell us about what pollutants or what impact the stream might be experiencing. A common thing in our field, environmental science field, is the importance of biotic indices and kind of telling us about the overall water quality of an area. You can go to a stream and take a water sample, and that'll tell you about the chemistry of that water at that point in time. But the fish, the mussels, the insects, the biota that live there year round, they fully experience the quality of that water body. And so they are a better indicator they are one indicator, but many people think might be a better indicator that tells you how good the water quality is. So we find certain fish are indicators of good water quality, and then other fish are indicators of poor water quality.
Excellent. Thank you, Dave and Allison. We do see that Cindy has her hand up, so I'm going to go ahead and stop sharing the screen. And Cindy, if you want to go ahead and ask your question. Cindy, are you there? No? Okay, not a problem.
I see Cindy's question though, and I think we can
yeah.
some thoughts about it. Cindy's in the chat here. We see that can we actually change the fate of the brook trout needing cold water during this time of global warming? What pollutants are the biggest challenge to fish? So I think one of the biggest, I'll just say that right now, I think the temperature is a really big factor for a large group of fishes. You have fishes like salmonids, trouts, that need cold or cool water to survive, to exist. And as the planet continues to warm and climates start to shift towards warmer climates, it puts those species at risk. And we're seeing that right here in the Mid-Atlantic and the Philadelphia region. And so the fate of brook trout, I'm an optimist. I like to think optimistically. A couple things that can help brook trout persist and hang on in this region are groundwater. So even though the temperatures of the region are increasing, spring-fed streams are areas where this fish can persist. Also, planting trees. A researcher here that I used to work with, Jerry Mead, did a lot of work looking at the value of planting trees in shade. And it's just something everyone here knows. On a hot summer day, you can walk under a tree and you feel the instant relief. This is the same thing for streams. So we can do this by planting trees along streams and in the riparian zone or the edges along streams to help shade and keep streams cool. That combined with groundwater, I think, are two good strategies to try and protect and conserve brook trout.
Yeah, I think along with that too, protecting our headwaters of our streams is also really important. So making sure those stay forested and protected and taken care of can really help the whole systems and give them habitat and areas to live.
I have a question. We talked a little bit about pollution, but if you had a really healthy stream or system, what kinds of things would you expect to see there when you're out sampling?
In this region, you have to say like.
Does it exist? Yeah, in this region. In this region, like what would you be looking for to indicate that it's a really healthy system?
So we have a lot of variety in the Delaware Basin. The easiest place to talk about right now, I think, would be headwater streams. Headwater streams in high gradient areas, the more mountainous areas, you would expect to find brook trout and sculpin, slimy sculpin. That would be the fish community that you would expect to see. And you would expect to see some other non-fish as well, so other salamander species. So I'll try and stay to fish, continue to talk about fish, but we also at times do studies that look at salamanders. And so some other salamanders would be northern dusky salamander or the spring salamander. These are good indicators of really good water quality headwater streams. And this is the assemblage that you'd like to see. As you come downstream, it becomes a little harder to answer your question because you start getting a lot more variables. I think one thing that we like not to see, let's go to the opposite end of the spectrum, is when we find areas that have a lot of black-nosed dace or green sunfish. We'll call them tolerant species. That's an indicator to us that the stream is really stressed and not functioning well. We'd like to see a mix of predators, invertebrores, and a variety of different fish species filling the various niches that you would expect. So as you come down to the Philadelphia area, you would not like to see a very simple fauna like I described for the headwaters, where it's just two species. Two species in the headwaters, brook trout and sculpin, that's what you'd expect. Down here, you'd expect much more variety. And so if you don't see that, that's a sign that things are not going well. Mm-hmm. The one thing I'd add before, just to close this one out, down here, we also expect to see a lot of migratory fish. People forget we're a coastal system. So we expect to see American shad and herring and American eel and striped bass and these fish that move. So good connectivity and fish that indicate good connectivity is something we'd like to see in our streams as well. And when we don't see that, that's a red flag.
I think we do have a photo that starts to show some pictures, Dave, of some of the species you're talking about. So I'm going to go ahead and share my screen, and perhaps you could also walk us through some of the other species that we could see as sort of our freshwater friends.
There you go. There's the northern dusky, and with the with the young there, which is that was a great find. I can't remember where we found that at.
Aquascicula.
Okay, Aquascicula. There you go.
Which
That's
is
up at,
headwaters, small stream there too. So lots of, as Dave was saying, lots of trout and sculpin at that stream. And yeah, the baby salamanders were a pretty neat thing for us to see.
yeah, our herpetologist, at the academy, he told us that was a great find. So the let's see what we have here. Some brook trout, tadpole, mad tom. All these fish you expect to find in different areas. Fish, habitat is very important. The sunfish you'd expect to find in lentic habitat or more pond habitats, slower flowing waters. The shield darter and the brook trout and the dusky salamander, you expect to find them in more coarse substrates, so more high gradient flowing streams. And when you don't find them where they're supposed to be, that tells us something about what's going on. When we find the sunfish in a flowing stream, that's okay. But when we find them to be the most common species in a high gradient flowing stream, a rocky babbling brook, that's a red flag. And that's one of the things Alice and I try and figure out and try and understand, so that we can help people make decisions on how best to manage the resource.
Dave, could you explain a little bit more on why it would be considered a red flag when you're seeing sort of a high population of the sunfish in sort of the babbling brook?
Yeah, let's say, for example, the sunfish, red breast sunfish, that is something you would find in a wadable stream around here. But let's say there's certain sunfishes that you would expect to find that would do well in a lake or pond habitat. So when you're in a stream, in a flowing stream, picture that babbling brook in your mind, you don't expect to find a lot of, say, the tadpole mad tom or the sunfish species. And so that's what I mean by a red flag. You find something that seems out of place.
Okay.
And so Alice and I spent a lot of time understanding the ecology of this area and the other areas that we worked, and the life history of the animals that live there, the fish that live there. How do they go about the course of their lives? And when you find things that are out of place, or you find that human beings have tinkered with something, we try to connect the dots.
Mm-hmm. You guys are like sleuths then, right?
Yeah, that's
excellent.
And Dave had mentioned it briefly before. Obviously our main focus is the fish, but we also do collect and identify and record the different frogs and salamanders and crayfish that we get as well. And those don't always go into all of our analyses, but they're also really important components, and they can help us understand kind of an overall view of what's going on. We can always go back to that data later. So pretty much anything that you would encounter in the stream when we're sampling, we'll collect and identify as well.
Excellent. Well, we are approaching our 1 o'clock at the end of our lunch and learns. These are sort of bite-sized little pieces of our science at the Academy. So are there any other final questions out there from our members? If not, if you haven't been able to answer or ask your question today, you feel free to email membership@ansp.org, and Allison and Dave would be more than happy to sort of answer afterwards. So Lindsay, are you seeing any more hands up or anything in the chat?
No, I'm not. I actually had one final question. I just was curious if you could take us through a little bit about after you get back to the Academy, after you've gone to the field and sampled, just to wrap things up, what do you guys do when you come back to the Academy with that data?
So sure, we're actually going through that right now. Now our field season is just about over. We were had a really busy last month and a half trying to cram our whole field season into just a short period of time. So we're back now, and it is all about data entry, checking over our field sheets, making sure, we got everything from the field and we're entering it into our database. It'll eventually get checked, and then we can start pulling it and start analyzing it, running statistics on it, hopefully either putting it into a report for a client or into a manuscript that we can hopefully get published. So right now it's all about data entry currently, and then hopefully moving on to analysis kind of in the winter months. And we also, too, for some of the projects, we bring back samples with us. Maybe if the fish are very small, they're hard to identify in the field without a microscope. So we'll be working through some of the things that we bring back with us over the winter as well. But it's all about the data in the off season.
Dave, is there anything else that you want to sort of add about what you guys are doing once you get back to the academy, or any sort of findings, that any hypotheses that you're thinking about that you should be looking out for when you move into the analysis section of your fisheries work?
I think the one thing I would add, before we even go out in the field, we go to great lengths to come up with our hypotheses and design our studies, and then the field work's all about gathering the data to help us answer those questions, which then tie back. Once we answer those questions, that's what helps move the ball forward in terms of making decisions for whatever the particular topic is. So that is also a very interesting part of what we do. It's just not, maybe it's not as interesting to talk about or show, but it's definitely part of the enjoyment that we get out of it. So.
Thank you so much, Dave and Allison, for your time this afternoon on this beautiful fall Saturday at the start of October. So we are so grateful. And thank you so much to our members who joined us today. And we look forward to hopefully seeing you this afternoon for our virtual collections tour with our paleontologist, Jason Downs. And then later this week, hopefully you're able to join us for our lunch and learn, specifically on our chemistry section. Dave sort of mentioned how we need to also analyze the chemistry of a stream to really understand what's going on in the habitat. So I hope all of you will join us on Monday for that lunch and learn, and continue understanding what it takes to have a healthy water stream and a healthy watershed. Thank you all so much. Thank you for your membership. With you, we are a force for nature, and our science never stops. Enjoy the rest of your afternoon, and we look forward to seeing you at more Members Week's activities throughout these next couple days. Thank you, and bye.